Ever wondered how monoclonal antibodies function and where they are applied? Let's dive into it. Monoclonal antibodies, or mAbs, are lab-created molecules that can mimic the immune system's ability to fight off harmful pathogens like viruses. They're made by creating identical immune cells that are all clones of a unique parent cell, hence the mono in monoclonal. Now, what makes these mAbs special is their ability to bind to a single type of antigen. Imagine each monoclonal antibody as a key designed to fit a specific lock, the antigen. This unique property allows them to target one specific type of cell or protein, making them invaluable tools in medical treatments and research, from cancer therapies to managing autoimmune diseases. And even in the fight against COVID-19, these mAbs have proven to be game changers. So, monoclonal antibodies are essentially our body's secret weapons against specific antigens. Now, you might ask, how are these monoclonal antibodies produced? Well, let's dive into it. The production of monoclonal antibodies is a fascinating process that begins with a humble mouse. The mouse is injected with a specific antigen, a substance that prompts the body to produce an immune response. This could be a part of a virus, a bacteria, or any other foreign substance. In response to this antigen, the mouse's immune system swings into action, producing a variety of antibodies, each designed to bind to different parts of the antigen. These antibodies are produced by B cells, each of which produces a single type of antibody. We then harvest these B cells from the mouse's spleen. Here's where the magic happens. These harvested B cells are fused with myeloma cells, a type of cancer cell. Why? Well, your average B cell has a short lifespan, while myeloma cells are immortal. They can divide indefinitely. When we fuse these two, we get the best of both worlds, a hybrid cell called a hybridoma that produces a single type of antibody and can divide indefinitely. These hybridomas are then cultured in a lab. Each hybridoma multiplies to create a colony of identical cells, all producing the exact same antibody. To ensure we have a pure sample, we separate these colonies, allowing each one to grow in its own flask. After a sufficient growth period, the hybridomas are stimulated to produce antibodies, which are then released into the culture medium. We then harvest these antibodies from the medium, purify them, and voila, we have our monoclonal antibodies. These antibodies are identical and specific to the antigen that was initially injected into the mouse. This specificity is what makes monoclonal antibodies so valuable in medicine and research. They can be designed to target specific cells or substances in the body, opening up a world of possibilities for disease diagnosis and treatment. Thus, we end up with a pure single type of antibody, known as a monoclonal antibody. We know what monoclonal antibodies are and how they're produced, but where are they applied? Well, they play a crucial role in the field of medicine, especially in the treatment of various diseases. Let's begin with cancer, a disease that has been a significant challenge for medical professionals worldwide. Monoclonal antibodies were initially studied for cancer treatment and are used today to treat certain types of this disease. Some of these antibodies are designed to target specific tumor antigens, stimulating a strong anti-tumor immune response. And even though some limitations exist, successful examples like the humanized anti-HER2 monoclonal antibody, Herceptin, have proven effective in patients with chemotherapy-resistant breast cancer. Now, moving on to autoimmune diseases. Rheumatoid arthritis, for instance, progresses with the help of a cytokine known as tumor necrotic factor alpha. Monoclonal antibodies effectively bind this factor, thus paving the way for a therapeutic solution for this debilitating condition. But that's not all. Monoclonal antibodies also come into play when treating infections. They have been used to generate antibodies against antigens resulting from gene mutations expressed by tumor cells. In practical terms, patient tumor cells are tagged with these antibodies carrying toxins or radioactive materials. This creates a direct, magic bullet therapeutic effect on the tumor. sparing the healthy cells. These antibodies also serve as a significant tool in disease diagnosis. They detect specific antigens circulating in body tissues using immunoassay techniques. In recent developments, the COVID-19 Prevention Network is studying monoclonal antibodies for potential treatments against COVID-19. So, as we can see, monoclonal antibodies have vast applications in disease treatment. They offer a way to target specific cells, making treatments more effective and reducing side effects. As research continues, we can look forward to even more innovative uses of these antibodies in combating diseases. Monoclonal antibodies have revolutionized the treatment of many diseases, offering more targeted and effective therapies. The future of medicine is indeed promising, thanks to the power of monoclonal antibodies. So, what does the future hold for monoclonal antibodies? Imagine a world where disease is less a threat and more a challenge, one we're well equipped to handle. This is the future monoclonal antibodies promise us. The potential for new applications of these biological marvels is vast and awe-inspiring. Scientists across the globe are tirelessly working, pushing the boundaries of what we thought possible in disease treatment and prevention. One of the most exciting areas of research is the potential use of monoclonal antibodies in the fight against COVID-19. Researchers are exploring how these antibodies can offer short-term protection against SARS-CoV-2, the virus that causes COVID-19. Clinical trials are underway and the early results are promising. But that's not all. The beauty of monoclonal antibodies lies in their versatility. They can be engineered to target a wide range of diseases, from cancers to autoimmune disorders, cardiovascular diseases to transplant rejections. They're not just a one-trick pony, they're a medical Swiss Army knife, ready to tackle whatever challenge comes their way. Let's take a moment to recap what we've learned. Monoclonal antibodies are laboratory-produced molecules, tailored to serve as substitute antibodies that can restore, enhance, or mimic the immune system's attack on disease cells. They're used in several therapies, aiding in the diagnosis and treatment of numerous diseases. They've been instrumental in the fight against cancer, using their magic bullet effect to target tumor cells while sparing healthy cells. Moreover, they've shown promise in treating rheumatoid arthritis by binding the tumor necrotic factor alpha, a cytokine that aids in the disease's progression. And now, they're being rallied in the fight against COVID-19, potentially offering short-term protection against this global threat. The future of monoclonal antibodies is bright, filled with promise and untapped potential. Their versatility, precision, and adaptability make them an invaluable tool in the world of medicine. As we continue to study and understand them better, we can only imagine the breakthroughs that lie ahead. In the hands of science, monoclonal antibodies hold the promise of even more breakthroughs in the future. They truly are the tiny giants of medicine,